All right, well, thanks for the introduction and the opportunity to come and talk today. So we'll continue our presentations on the challenges, but as we said, we'll start off with the chemistry side of things, and I get a laser pointer. So I've broken it 4 different areas. Going to talk a brief discussion over the chemistry data recommendations that we like to see in a food contact notification. Again, like Jason, I'm going to talk about some of the common deficiencies where I'll kind of compare and contrast what we've seen and what we're kind of looking for, as well as talk about some of the new challenges, and I'll address more specifically the migration conditions for the packaging materials used in contact with infant formula. And if I've got time, I'll talk a little bit about the series knowledge base and some of the work we're doing with that system. It's been a while since I gave ILSI an update on that one. All right, so this slide actually breaks down the 9 different data recommendations that we have for the chemistry side of the food contact notifications. It really can be simplified into what is the food contact substance, what materials have the potential to migrate to food under the intended conditions of use. We're looking at what the food contact substance itself, as well as any of the impurities or breakdown products that might be generated during manufacture or use of the material. How much of those things are actually migrating from, say, a packaging material into food, and human exposure to that material. Okay, the next couple of slides I'm going to kind of touch base on a number of these different data recommendations here. So one of the first things I want to talk about is the identity of your food contact substance. We really would like to have a very concise identity. Okay, there's no secret food contact materials, so we really need to have a very specific name for the material. So we're looking for a systematic name, a chemical abstracts name, or number. We've had a few people recently put in things that are very generic. Alkyl acrylate this, alkenyl acrylate that. You can imagine it could be any one of several dozen materials, so we really need to know a specific identity for your particular material. The other thing I want to touch base on is the use of your material. It needs to be clearly defined. Okay, the use is actually going to drive pretty much everything else in your chemistry section of your food contact notification. Okay, it'll drive what types of migration studies you need to do. It'll also drive the exposure scenarios that you might use. Okay, so if you're using a repeat use item, okay, we have specific exposure scenarios for that. So we're looking at residue levels, and you can use the quantity of food that that particular repeat use item will contact over its service life and derive a dietary concentration from those. Those things are usually fairly small exposures, as opposed to a single use item, such as a packaging, my can here. We have a number of different time temperature conditions, A through H. I think some of you are aware of those things, as well as the different food types. So the use is really important because it's going to drive the rest of your information. The other thing I want to draw out is manufacturing information. Really need a complete manufacturing process. Okay, times, temperatures, pressures, all the reagents, okay, that are going in there in the quantities, as well as any purification steps. Again, this drives your migration experiments as well as exposure scenarios. It's no sense in looking for diethyl ether in your migration solution if your last step of your manufacturing process is a purification process where you're distilling something at high vacuum and high temperatures. Okay, so we can use that information to do some scientific rationale as to why maybe this material wouldn't be present in the final product. Okay, so that drives some of that migration experiment in exposure scenarios as well. Actually, one of the biggest things that we have a problem with is supporting analytical data. We really need that supporting analytical data for all of your analytical measurements, because we actually go back and run through all your different calculations, recreate your calibration curves, look at the validation process, and take your raw peak areas and run that all the way through to concentration in food. So we're really looking at give us all the raw data for your analytical measurements. We want sample chromatograms. You don't have to give us all of them, but sample chromatograms for all your different measurements. Calibration curves. And make sure the calibration curves actually match, so I can take a raw number, peak area, and run it through the calibration curve, and I'll actually end up with the concentration in the food that you're actually claiming that you calculated out. It's important. I've seen calibration curves come in where it's a nice calibration curve for the right material, but when you actually do the analysis, nothing kind of works out in the end. So make sure the calibration curve is the one that you're using for your particular experiment. Validation data, we have to know that this analytical method is going to detect what you say it's going to detect. And I don't know if Tim will be talking about validation. I know we've had some issues with some of the perfluorinated materials that seem to stick to the glass containers that you put them in, so there's different protocols that you need. You have to be able to validate and show that you actually can get the material back out once you spike it in there. The other big thing I think Jason talked a little bit about is narratives. Okay, we'd like to see really nice narratives for each of your analytical methods, and then a narrative of how you're going to do your exposure assessment. Okay, so we need to know, did you do dilutions? If you did, what were they? So we should be able to follow all this through the whole process and come up with essentially the same answers that you guys did as well. So we need a good narrative. Tell a story. One of my colleagues, Alan Bay, likes to tell a good story on this information. The guidance documents, all the things that we talked about here, all the things that Jason talked about, and probably what Paul Honigford will talk about later on today, can be found in our guidance documents that I've got listed here. If you still have questions about how you might want to conduct a migration study or how you might calculate exposures, we do have the pre-notification consultation process. So this is a tool that assists you guys, the notifiers, through our regulatory process. Okay, it can be done in any form, written, so you can send us a letter, you can email us, you can call us on the phone. We're happy to talk to you, or we can do in-person meetings or WebEx meetings. This will cover anything from clarification of regulatory status of a material, if you're interested in a cumulative estimated daily intake or an ADI value, all the way through discussions on alternative approaches to determining exposure, as well as safety. Jason talk about read across and things like that, so you can come talk to us. Even have people submit fully formed food contact notifications and ask us to go through them and point out any holes that might be in the submission, and we'll provide feedback on that. So we really want to actually help you through this process. So when you come to us, make sure you give as much information to us as possible so we understand what your material is, the intended use. Okay, I'll harp on that a little bit, so we know exactly what it's being used for, how much is being used, because that's really going to drive our recommendations for the types of migration studies or how you might calculate exposure, which then is going to cascade down to, Jason talked about the tiered toxicity testing recommendations. So we really need that. That's going to follow through on that. So that's the notification, pre-notification consultation process. As Jason pointed out, we have some new things coming down the pipe at us. He gave you a little bit of an input on the food contact substances that are going to be used in contact with infant formula. I'll take the next step, and we'll talk a little bit about the migration protocols. So we've laid the migration protocols out, as you see here on this slide. So for liquid formula, so that's ready to feed and concentrate, we're looking at 50% ethanol. Okay, conditions of use would be A or B, depending on the particular container and its maximum useful temperature. Powdered infant formula, you could do 50% ethanol. It will exaggerate migration, but you could use a dry simulant such as 10X, as well as the conditions of use E. Okay, for articles that are used to feed infants, so we're talking about baby bottles and nipples and things like that. These are repeat use items. We do recommend a 50% ethanol food simulant under conditions of use B. Because it's a repeat food simulant, we calculate exposure a little bit differently. Instead of using consumption factors and food type distribution factors, you can look at looking at the quantity of food that will cover each square inch of the baby bottle or the nipple, and we've just developed some numbers for that. As Jason pointed out, the dietary intake for infants is a little bit different. It's more like a direct food additive or grass ingredient, so we're looking at use of no consumption factors and no food type distribution factors in this. Okay, we're looking at using the residue level, whatever your material you're interested in looking at, times the quantity of food that an infant ingests per day. And then we can divide that out by the infant body weights that Jason was talking about there. So you end up with an estimated daily intake, no dietary concentrations or anything like that, straight to the intake. Some of the other things that we've been working on are changes in the way we think about things is we're getting different types of submissions for different types of materials in. This is an interesting graphic that I had one of my people put together. This is actually the number of submissions for antimicrobials that we've received in our division since the 2000 all the way up to the present, somewhere in August, I think, of this year. You can see a huge uptick in the number of submissions for antimicrobials that are being developed for use in processing of food, such as meat and poultry, and even fruits and vegetables. The other things we see coming in is a shift in the marketplace due to either consumer demand or other reasons out there. So we're looking at different types of materials. Can coatings are changing because of bisphenol A and the consumer demands there, as well as the types of submissions that are coming in. Our office is primarily a pre-market side of things. So for food contact notifications, we get about 100 food contact notifications a year. It's been pretty steady for the past, 10 years, I think, so we get about 100 of them. But we've also, in the last 2 or 3 years, been getting more post-market submissions to our office. Okay, food edit petitions to request the removal of a particular material from the Code of Federal Regulations due to potential safety issues for that particular material. We're also getting submissions in from industry and groups like ILSI and others that are requesting to remove things from the CFR due to abandonment, okay, which is a different shift for us, and we're really actually interested in working with you guys as part of product stewardship or chemical stewardship of actually going through the Code of Federal Regulations and removing those compounds that are abandoned. So if you guys want to actually talk to us about abandonment of chemicals and things like that and submitting a food additive petition to get those off the books, we're happy to talk to you. You can set up a pre-notification consultation for something like that. Because we're seeing a shift in some of the different types of materials that are coming into our office, we have to look at our consumption factors and food type distribution factors. We actually had 3 interns in our office. They were looking at the food label and packaging survey, or the FLAP survey, which is collects labeling information on our FDA-regulated processed and packaged foods that are marketed in the United States, and it's really meant to monitor industry's response to labeling regulations. But what's really kind of cool for us is that they actually buy products off the shelf, they clean out the insides of them, take whatever data they need, and then they actually ship us the packaging material. So we've got 3,800 cleaned packages sitting in our office, and our interns actually went through that with the help of Tim and some folks in his lab, and we actually analyzed and looked at the food contact layer of each of these packaging materials, and we're hoping to take that information along with market survey data such as the Mintel survey and refine or update our consumption factors and food type distribution factors. Okay, so this is another area that if you're interested in talking to us, and if you have data that we could use to update our consumption factors and food type distribution factors, that would be great that we could partner and do that. Because of all this changes, we're updating our chemistry guidance document, so we just started working on that. I just finished reading the first draft of the first section of it here this past week. It's going to be reorganized, so it's actually going to follow the format of the dreaded Form 3480. We'll have updated references, discussions on stability testing and food simulants. One of the big things we're putting in here is updating a little bit more on the migration modeling, especially cooking applications, other calculations for specialty scenarios like wet end, paper additives that are using the wet end of the paper manufacturing process, things like that. And the other big thing that we're putting in there that has never been in there before is the development of market volume limitations. So what kind of data you need to submit for that, and how you do the calculations for that. So that's a way of refining a consumption factor for your particular product and your particular market. The other thing I wanted to talk about is the series knowledge base. It's a food additives knowledge base that we've been working on in our office for I forget how many years now. And it's actually moving along. It's gained quite a bit of traction in the office as well as the center. It's a chemical-centric database that captures our institutional knowledge consolidate some of our internal databases. Everybody has internal databases, all these little access databases or spreadsheets. We're trying to pull all those together in one source and link series up to some of our other big databases within the office and the center. Okay. It contains about 90,000 unique chemicals. Okay, covering grass ingredients, food additives, food packaging materials, colors and cosmetics, dietary supplements. Of those 90K compounds, roughly 46,000 of them have structures that have been curated against chem abstract services. So we have pretty good structures for a lot of these materials. It links that information to our regulatory side of things. So where have we seen this before? Okay, linking styrene to particular food additive petitions, food contact notifications, subject files that talk about styrene, for example. Okay, human intake values are in there as well. And we're collecting a lot of toxicity information to go along with that process. So we have 32 different tox assays, both in vitro and in vivo assays. So right now we have here, about 13,000 tox studies in there. Okay, so we've actually collected most of the direct food additives from the PAFA database, the food packaging materials, and right now we're just starting the collection process of the, I don't know how many hundreds of indirect food additive petitions that we have. So we haven't touched those, so there'll be quite a bit of data in those we anticipate, so we're going to go through and start collecting that information. You can search series in a number of different ways. You can search it through names, chem abstracts, numbers, other identifiers, structures. You can do structure similarity searching, substructure searching, exact search. So we can start doing that read across. We can start doing that structural analog searches. You can combine that with a search for specific toxicity data if you're interested in it, or just look for toxicity data by itself. Give me all the carcinogenicity studies that are Ames positive or Ames negative. Okay. Other things that we're working on, Jason touched on a little bit, is developing some QSAR models that are inside the series system, so you can use series as a workflow. What do we know about a compound? Okay, maybe we don't have a lot of tox data on it. We can move to the next process. Let's run through a QSAR model. Okay. We've got export features, so we can dump our data out to incorporate it in memos. We got compared structures. This is kind of a neat little tool that we developed. So if you have a query compound and you want to do your structure or analog approach, you can actually put your analogs in and get a numerical score of how closely they match each other in a couple of different metrics on that. And there are a number of different things that we're working on, TTC being one of them. So we'd like to develop the new decision tree that Tim Adams talked about, I think, here at one of the SOT colloquia we had at our center. So we're going to develop a module for the series system to take advantage of all the work that he's been doing. As I mentioned, institutional knowledge, big thing. You guys send us all these notifications. We generate a lot of data when we review them, human intake values, new no-Ls, low-Ls, study calls, et cetera. So what we're trying to do is develop some tools that allow us to capture all that information right in our workflow process and stick it into the series system. So we've got a tool that we can register the chemicals and the structures, so we can have curated structures right up front and curated names. We have a tox data entry tool that we're going to be building that allows to capture that the old legacy data from the old food additive petitions. And finally, we have what we call my review memos. Okay, these are review templates that people can use in their regular review process and allow us to capture the information that they're generating and dump it right back into series so it's available for somebody else. We get a lot of repeat submissions in our office, so we'd like to be able to grab that data from something we've seen before. We look at it, do we need to update it? If we update it, grab that new information and put it in, so we have this nice cyclic process of updating the series system. As I mentioned, we want to be able to actually share the data with other systems, make sure we're all talking about the same stuff, same IDs, same structures, et cetera, so we can find our information a little bit better as well. Am I doing okay on time? Mm-hmm. All right. Oh, okay, I'm going to rip through this here. We've also done a little bit of work on some of the ToxCast, Tox 21. One of my staff fellows. Patra Volaroff has actually gone in and looked at the overlap between ToxCast, Tox21 chemicals, and the chemicals within the series knowledge base, and have kind of got that summarized here. So you can see we actually have a pretty good overlap. So if the ToxCast chemicals, about 86% of ours overlap with, or we have that overlap, and with the Tox21, it's almost 93% of that overlap. Some of the future directions for Ceres and Office, we've been working with the Flavor Extract Manufacturers Association. We actually have their data that we want to put into the series, so we can look at that when we're doing evaluations. Always looking at to improve our QSAR models, how to integrate high throughput screening information into our review process. New tools that allow us to do some of the knowledge gathering, as well as some of the read-across type tools in TTC to support our post and pre-market activities. If you have any questions about Ceres, you can come talk to me. It'd be great. Thank you. If you have any questions, happy to answer them. I probably forgot something.
Thank you, Kirk. Any questions? Up here in the front. Oh, right next to each other. Perfect.
Sean gets to go, I guess.
Thank you. That was a very useful presentation. On the question of migration modeling, and you said you're going to provide some guidance. One of the challenges is that there's a number of different models available on the market, both free and commercially available. Are you going to address that question of the appropriateness of use of the different models that are available for different applications?
Yeah, we'll have to address those things. Right now, we actually have access to the MigraTest software in-house, that we've been using that. And Tim's pretty familiar with the migration modeling as well, so we'll have to think about that. It's very similar to the QSAR process, which models to use, and one of the things that we've, I'm head of the QSAR team, one of the things that we did with the QSAR models is that we collaborated with some of the big names. So we have the LeadScope, Multicase, Plaza, Derrick products. So we actually have kind of a breadth of systems. So if somebody submits us something, we can actually rerun it and check their information. So that's kind of one way that you could kind of address some of that, is have multiple systems available, so we could recreate your migration experiments.
I had a question about consumption factors and whether you consider things like the dietary intake in terms of caloric intake of different types of foods that would be in packages to get more realistic estimates of exposure, because if you assume that total dietary intake from a package that's used exclusively or for one group of foods, if you assume the grams of intake, it may be so grossly overestimating what's allowed to be for caloric intake in somebody's diet that you get really unrealistic estimates of exposure. That's one. And the second is, have you considered the intake or exposures from other FDA-regulated types of products, such as cosmetics, et cetera, to say, where is the total body burden for these chemistries that you're looking at, and looking at total exposure instead of just from the dietary route alone?
Mm-hmm. Mm-hmm. That's a good question. So yeah, the caloric question, that kind of cropped up a couple of different times with respect to how to calculate exposures with respect to like infants and things like that. So, that is something that we need to think about when we're going through and looking at our consumption factors, and see if we need to incorporate something like that in there. With respect to looking at the different sides of the coins with different exposures, that is something that depending on how we have to look at it from a legal standpoint, we may have to consider other sources. I know, for example, when you're doing direct additives, there's been some petitions in for vitamin D. So you have to look at what's already in foods with respect to vitamin D, what's naturally occurring, how much they're going to put in now. So we do take those things into consideration when necessary. Mm-hmm. Yeah.
Over here.
Could you touch on the major differences between the FCN process and a GRAS type of certification? If you want to get material under the generally regarded as safe, kind of how big are the differences in the process?
Oh, well, that's interesting because I'm actually doing a 90-day detail in the division of biotech and grass. Notice I've only been there for like 3 weeks. So, but yeah, there's a difference. The biggest difference is that the food contact notification program is mandatory. You have to come to us, and we have a mandatory 120-day deadline to review that. Okay, so you have to come to us before you market your material. The grass notification program, it's a voluntary program. They're coming up with some of the new, the final rule is available for your review. They're putting in some timelines for that. I think it's 180 days for their review, but it's not really a statutory hammer like we have in the food contact notification program. From the side of the chemistry and the toxicology side, we have the same safety standard across all of the programs that we have in the office. So food contact substances, the TOR, threshold of regulation exemptions, the GRAS ingredients, as well as the direct food additives and color additives, they all have the same safety standard. And you're really actually getting pretty much the same types of data in. So we need to be able to generate, what is the material, what is it going to be used for, how much people will be ingesting with it when it comes on the market, and then, is it safe for that intended use at those use levels. Okay, so it's pretty much the same. Dennis wants to chime in.
Yeah, this is Dennis Keefe. I'm at OFAS. Just another distinction between the GRAS notification program and the food contact notification program. There's exclusivity with the FCN program. There's the FCN program. The result of the FCN program is actually what's considered a decision by the agency and an action. So there's a NEPA component to the FCN program that is not part of the GRAS notification program. So there's an environmental obligation as part of the food contact program. That and exclusivity, the hammer, or the timeline, the 120 timeline is the real difference between the two programs. I should say also that, in terms of the food additives program at FDA, the food contact applications historically has been the greatest amount of our work. So if you look at the number of approvals or effective FCNs, that is, over the history of the program, that is what the office has really been doing in terms of the widgets that we produced. It's not the direct additives. I think that's something that might be surprising to most people. But if you look at the number of approvals, and I put approvals in quotes, it's really for food contact application that the office has been involved with.
Well, thank you. It's time for us to move on. So thank you again.